Okay, so good morning, everybody. Welcome back. Stefan, first of all, thank you for coming. Obviously, you've been quite a busy person in the past year, so I appreciate the time you're making here. For the audience, it would be great if people could feed us questions during the discussion. Don't wait for the end in the Q&A, so that if we see some relevant questions to the topic that Stefan and I are discussing during the fireside chat, I might be able to give some of those questions to Stefan. So please send us a question in real time. I can see them right there on the screen. So, Stefan, let's just start with the chronology. I mean, of the year in review, but you've covered that in about a dozen panel, maybe more than that. So let's not give the long story. I'd like to focus on, with you not thinking too much about it, as you recall that year, which is probably kind of a blur in some ways. What are the key inflection points, not just for the company or for the field and the overall vaccine effort, but for you personally as well? Like, what are the moments that you will never forget, that you kind of remember crystal clear?
Great. So, JF, thank you so much for the invitation. It's great to see you. So I think last year started first at the end of 2019 for me, when I first heard about this new virus in China. Didn't know what it was. I thought it was going to be an outbreak like MERS or SARS. So one of the first pivots in the year was my week in Davos 2020, where I walked into the World Economic Forum thinking this new virus that at the time still had no name was going to be like SARS or MERS. And I left Davos convinced it was going to be a pandemic like 1918 flu. And I changed my plan. I was supposed to go to QIAGEN board in Germany. I took a one-way Zurich to Washington, D.C. plane, to spend a Sunday on a plane to meet Tony Fauci Monday morning, to go to the FDA in the afternoon, see Marion Gruber, who run the vaccine division, and see everybody, BARDA, DARPA, and so on that day, because I was like, this is going to be really bad. And then we're just chasing the virus. We're starting to think with a team, how, if a vaccine works, can we get to 1 billion doses next year, which was a crazy adventure because we had made less than 100,000 doses in 2019. So it's kind of 10,000x increase of capacity. And so we spent all the spring working on the clinic, working on manufacturing scale up. And then the issue started to be money, which is we need billions to be able to build that manufacturing infrastructure. We don't know if a vaccine is going to work or not. We've asked governments, nobody's giving us any money because we have no data. And so on May 18, when we basically get the neutralizing phase 1 antibody out, which are very strong for all the participants, we raised that evening after the market closed $1.3 billion. And Morgan Stanley took the entire deal, and they took the deal on the bank's balance sheet. Because James Gorman called me in the morning. He's like, you don't have time to do a roadshow. I say, I would really have to not do a roadshow. He's like, well, we'll do it. We'll help you. We'll do. So they take the whole deal at 401. They buy $1.3 billion of stock, which was a lot. And then we scale up manufacturing very, very quickly, get into the phase 2, the phase 3. The phase 3 starts end of July. And one of the biggest decisions and hardest I had to make last year is to decide to slow down the phase 3 around mid-August, because we're tracking fantastic for, white participants, but the African-American participant and Latino were below where we wanted them to be. And we basically decided to slow down the study, to shut down sites that were recruiting too many white participants, to make sure we are meeting quotas that were similar to the national level quotas. And we're super happy at the end that we were successful, where we got 47% of the participants of colors across all the different demographics. So that was a tough one. And then, of course, Pfizer data coming a week before us, at 90+% efficacy. Having that Sunday debrief by the DSMB, where they basically tell us the vaccine is 95% efficacy. And
so you still remember that? You still remember that one, right?
Yeah, and I remember my entire life. I remember I was in my home office on a Sunday when Tony Fauci is on the call too. He doesn't have the data yet, and they are debriefing us. And they start talking, and it feels like hours and hours. I'm sure it was just a couple of minutes. And then they drop us the number. And I remember Tony saying, this is unbelievable. And then us, focusing on getting the EUA ready.
Yeah.
And if you think about the company, I'm so thankful to the team who has done just incredible work. I mean, in January of 2020, we had never done a phase 3 in the company history. We had never got a product authorized. We've never, manufactured at commercial scales. And, we did our Q1 numbers. We delivered the $100 million by the end of March, which was our commitment to the US government. And we're on track for, between 800 and $1 billion this year.
Hey, Stefan, just jumping it backtracking a little bit on the phase 3, the hard decision, right? So I hear that it was a group decision, right? I mean, to what extent was Monsef and Warp Speed or the FDA, right, were kind of part of that decision, and there was a back and forth, right? I mean, how did you guys get to the decision and the process, right?
Yeah, sure. So the FDA was not involved. It was indeed Operation Warp Speed because the US government was paying for the study. And so we had a couple back and forth with with Monsef and General Perna and Stephen Hogue, who is the president of the company who runs R&D, and I, about do we need to slow down or not. It took kind of a week going back and forth. And then they're like, look, at the end of the day, you have a sponsor, you have to decide what you do. You own the IND to the FDA. And we're like, look, we want the vaccine to be used. If we get the vaccine approved and only white people use it, we will not feel good about ourselves. And so we say, okay, let's take a slow down. It might be just a couple of weeks slow down. Yeah. It might be the right thing to do because we need to get the country to get to herd immunity, and we need as many people vaccinated as we can.
Yeah. So stepping back even further, at the very beginning of the effort, right, when I guess January, you guys go full speed with making the vaccine. Tell us a little bit about how much the NIH, was brought to the picture, right? Okay, I know we hear that they were very prepared. They've been working on some of those vaccines for years, but how exactly did it happen? Like, what did they have? Was it really ready to go, or was it kind of a back and forth for a couple of weeks or days, or?
So the background is quite interesting. So we have been collaborating with Tony's team for a few years before the pandemic starts. And every September, I go down to the NIH to look at the update on all the scientific data and so on. And they have been working on several vaccine candidates, and one of them was MERS, the Middle East Respiratory Syndrome, which, of course, as everybody knows by now, is also a coronavirus. And they had tried, like you always do in vaccine development, a lot of different antigen of a virus. They try combinations. They try, truncated part of a spike protein because it's such a big monster. It's a kilobyte message, and the full length spike. And what they realized, and they tried recombinant protein, DNA vaccine, and mRNA vaccine from Moderna. And in every case of what they tried, they saw that in challenge model, the best vaccine was always full length spike with mRNA. And so after that meeting, we talked about CMV and a lot of other things. At the end of the day, we talked to Tony about something that I've always been passionate about, which is, outbreak and pandemic readiness, because I knew the mRNA technology could go so fast. And so I say, hey, one day we should mock up an outbreak where you guys send us by email a sequence of a virus you pick, and we start the clock watch, and we see how quickly we can give you a GMP dose to get into the clinic. You guys take it to the clinic. And I told them, we should be able to do that in less than 60 days. And they were all laughing, saying, you cannot do a GMP product in less than 60 days. I said, yeah, we can, because we're already doing it for cancer patients, for personalized cancer vaccine. And so fast forward, they come visit the plant in Norwood in November. They talked to the team. I was traveling. I was not there. And then after they called me, say, yeah, we should do that in Q2, because in Q1, this is a true story, we should do the outbreak simulation in Q2 because in Q1, we're too busy. Okay, we'll do it in Q2. We have time. And then you connect the story. Then in December, I hear about the virus. When I hear about it, I email their team right away. And they told me, look, we've heard about it, but we don't know what it is. A few days after, we realized it is not flu. It is not MERS. It is not SARS. It's a new coronavirus. We only got the sequence, I think, January 13. And what happened that day, what we did is we had the NIH team design a vaccine in silico, and we had the Moderna Cambridge team design a vaccine in silico without talking to each other. Because what I wanted to make sure is that to see what they were recommending to do. And both teams came back after 48 hours of a lot of analysis, 3D simulation of protein structure and so on. both groups came out to Tony and I saying, we believe a full-length spike protein of the SARS-CoV-2 is the right vaccine to go after. So we said, good. We click order. He went to the robots to make both preclinical product, clinical product in parallel. And as you know, March 16 is when the phase 1 started.
So it's a good segue in the one of the most important aspects, at least in my opinion, of the old COVID vaccine stories, the public-private partnership, right? And love to hear from you. What I heard from Monsef. I spent a bit of time with Monsef, after in his post-warp speed, new life, but and he was telling me that there was very little interference from the White House, at least with warp speed, and at least he felt comfortable. A part of it, we won't comment on what he said about Trump, but he was talking about Jared Kushner, that was actually pretty, open-minded and didn't want to interfere because he felt that those guys knew better. What did you think worked really well, and what do you think that needs to be really thought through for the next time around?
Yes. So first few things. Collaboration with NIH, amazing. They're developing some assays, we're developing some assays. The team talking 3-4 times a day. So on the scientific front was fantastic. The FDA has been amazing. I've been in this business for 25 years. I've never seen what I saw last year. I never thought I would see what I saw last year. Sometimes we'll have 2 or 3 meetings in the same day.
Wow.
Where literally the team will get on a Zoom together, problem solve for a situation. FDA say, look, give us a few hours. We need to align internally. They'll go away. They'll send an email or give a call and get on the Zoom again. And in the same day. And it will be Saturdays, Sundays. I still have, in my folders, email at 2:00 in the morning, Saturday night. People work really around the clock. So that went fantastic. OWS, I don't know who thought about OWS, was a piece of genius because putting somebody from industry as a counterpart to us, who could in a nanosecond say, yes, I understand your problem, I'm going to help you, which is what Monsef did. Teaming him up with somebody from the military, who's kind of really good at supply chain and making shit happen and moving things around, was fantastic. Monsef was a blessing. Literally, he and I spoke at least once a week at 4:00 in the morning. Both he and I are early riser. I will text him and say, hey, I need to speak to you about something. In the next 30 seconds, he will call. We will align 5-10 minutes on something and get the day moving. So they were really fantastic. The one thing I wish the country had done better is giving industry financial support earlier. If you think and you play back through history, it's only until that, act passed in Congress that BARDA got any money. But we wasted precious time. I believe we could have had more vaccines in December if we got funded when I asked for it on January 27th.
So that actually, it brings thinking about biopreparedness, right? And how do we prepare for the next one? Monsef was reminding me, you remember that like 5-6 years ago, he and GSK approached the US government and European government. My understanding is that they had actually found a couple of plants that were older plants that could be repurposed. And they had put a budget, I think it was called Biopreparedness Organization or something like that. And apparently the US government and the EU government actually, turned them down. I mean, they didn't think it was a bad idea. They just were not willing to support it at that time. Do you think that that's going to change this time around? And do you think we'll see that kind of initiative?
I think it's going to change. But I think what's going to change the most is the fact that now you have mRNA capabilities with crazy industrial scale.
Yeah.
And we keep improving the timelines. I mean, look, last year it took us 43 days to go from sequence to shipping GMP product to the clinic. We did a South Africa variant earlier this year in 30 days, 3-0. I think we can still do a bit better. And so, unlike in the past where you had nobody with mRNA capacity, we've announced 2 weeks ago, we are adding manufacturing capacity because of the adenovirus vaccine issues and the protein delays. We're going to have 3 billion doses of capacity next year, 3 billion. And we have a flu vaccine coming, we have a CMV vaccine, we have 9 different vaccines coming. And so I think the world is going to be, if there is another outbreak of pandemic, in a very different situation. We're also working with governments, like we have a Zika vaccine going to phase 2. It's funded by BARDA. We're going to take it to the finish line. because the world needs a Zika vaccine authorized. I don't know when Zika is going to come back, if it's next week or in 20 years, but Zika is coming back. It has not left the planet, like, Ebola or the viruses. And so I think we can do a lot of work by getting ahead of those viruses, because then if you have a new mutation, a new variant of that virus, then there's a very clear regulatory pathway through neutralizing antibody levels with hundreds of people, not thousands, when you can get to a finish line. So I think there's a lot of learnings from where we are and technology making that I don't think next time will be as bad as this time.
So that's a great segue to the IP waiver and some of the compulsory license discussion within the TRIPS agreement, and all the things that have popped up in the past 2 weeks. I think it's probably easy to understand for most people why all the limitation, right, of the IP waiver and why it's probably relevant in the short term. But thinking at steady state and also for the future, right? Some 3 billion doses is massive, and I'm assuming Pfizer will have the same thing. But do you think the case can be made for further capacities that might be kind of a public-private partnership where you could actually manage this whole issue of IP in a better way, as opposed to just put it out there in the wild, where you know that it's not going to do much? So that would be the first part of my question. And then bear with me, the second part is, do you think that the argument for to support the limitation of the IP waiver for mRNA technology, given the complexity, we all know the supply chain complexities and know-how, do you think that they apply equally convincingly to other vaccine, particularly the coming recombinant vaccine, right? Once, let's say, Sanofi GSK is online, and I'm sure they won't like the IP waiver either, but some of those arguments may not apply the same way to some of the other vaccines. So, but let's start first with this whole discussion of IP waiver, whether you think that once we get a steady state, the same argument can be made for the limitation of the IP waiver.
Yeah, so the IP waiver for me has two dimensions, a short-term and a long-term dimension. I think, like everybody in the industry, the IP waiver is an incredibly bad precedent, in the sense that if there had been an IP waiver 15 years ago, for example, Moderna and BioNTech might not have existed, or might not have existed to the same scale. And think about where the world will be now, because, of course, investors need to get a return on the investment, whether it's a molecule in a big pharma company or biotech investment, as you know very well. So I think the IP waiver as a one event is a very dangerous slippery slope in terms of unintended consequences in the long term across the industry. The IP waiver, in my opinion, is not going to add anything to mRNA vaccines in 21 and 22.
Sure.
Because you have to set up manufacturing to scale, to get the product registered, to qualify the equipment, and so on and so forth. You have a raw material, as you said, and then you have a know-how. You cannot go and find on the street people that have 20 years of experience of making mRNA vaccine, because they are not. And so the whole IP thing, I think, is just politically most probably interesting for some states to say that they are willing to help and so on, because it's not going to help anything. It might be different, of course, for adenos or protein, because yes, for those there is existing manufacturing capacity, which is not for mRNA. There's no plant in India or China or Africa that has even 100 million doses of mRNA capacity available. This does not exist, and so it will have to be built, which is not the case, of course, for protein or adenos. But if you look at the maths, the issue is starting to be more and more raw materials, not vaccines. I mean, if you look at the capacity that's available today, look at what AstraZeneca has done. They have a massive ramp up. J&J is doing the same thing. If you look at the Serum Institute in India, the CEO has been very clear. He's not missing capacity or people. He's missing raw materials. Because in the US, we've had, Defense Act and all the rated orders on raw materials that we cannot ship. I have raw materials sitting in the US, sometimes raw material waiting in Europe to do products for outside the US. I'm not allowed to ship them. It's illegal.
Yeah. But so let's talk. So let's touch a little bit on the recombinant, for example, right? So is there? You can see where some people say, well, IP waiver makes sense here because there is capacity, there is expertise, but at the same time, the manufacturer will probably be ready to address the need within, let's say, less than a year for sure, right? I mean, Sanofi is moving into phase 3, right? You probably saw that yesterday. Novavax, I'm not tracked Novavax, but I'm probably looking at Sanofi and GSK more closely. Obviously, as you said, AstraZeneca, J&J are ramping up. There is one dimension that I'm curious about, which is right now you're selling to the government, right? I mean, we're under the emergency use authorization. How does that dynamic change a little bit once you kind of get approved and you start selling to customer? And tell us a little bit how you see the field moving once people gets approved, and what's going to be maybe the different commercial strategy and some of the issues that may come out of that.
Yeah, so, and I think that's mostly a US question you're asking, Jeff, because as you know, outside the US, government's buying anyway. It's the only buyer, in Europe or in Asia and so on. And so I think what's happening is we are moving into a boost market, because by the time the authorizations come, is going to be months from now. As you know, we are swimming in vaccine in the US. There's more, supply than demand. And so that's not going to change. My math is there's around 70 million doses in the US in the supply chain right now. So anybody who wants a vaccine can literally log on CVS, get a shot today in Massachusetts. You can play with CVS website and test for yourself. And so we're swimming in vaccine. And so the question is not going to be prime series, because anybody can get a prime series. The question is going to be boosting. As we've been saying for a long time, we believe people will need boost shots. We believe we're going to need multivalent, variant-specific booster, because the virus keeps evolving. And this is where the market is evolving. It's going to be a single-dose booster. And I think it's going to be about how good is your booster, how many strain can you put in the same dose. We are testing, 2 mRNA per dose right now. The existing authorized vaccine to boost all your antibody repertoire, but also the 351, South Africa strain, full mRNA spike in there as well. We've shown in humans data last week showing very good protection against not only, 117, the UK strain, but P1 Brazil strain, 351 South Africa strain. We're going to share Indian data very soon. But I could see a world, JF, where in 6 months from now, you might have 3 or 4 mRNAs in the dose that you get at the time you get your booster. That's why I think where the market is evolving. I think mRNA is going to have a big advantage, because when I hear governments, and I get, unfortunately, way too many calls from them those days, is people are saying, look, we want mRNA vaccines. And they say it's very simple. mRNA vaccines have the highest efficacy of all the class of vaccines. They have no safety, very good tolerability profile. You've had no manufacturing issues scaling up, because both Pfizer and us have delivered to a commitment in terms of delivery of manufacturing. And you're able to go very fast onto variants. We are going to get into a world very soon where we can go at Moderna in 100 days from a new variant of concern identified to an authorized product.
But Stefan, do you think that government demand does it mostly apply to the Western world? Because you guys still have the distribution challenges, right? I mean, I know they've been solved in many ways, at least in the US and Europe. But for the rest of the world, is that continue to be an issue?
It's not an issue. I mean, we have in a partnership, with COVAX and UNICEF for the poor countries. We have countries like the Philippines and others that are using our vaccine right now. And the vaccine, if you want, can be stored 1 month in the regular fridge. So what those countries do is they have already for other products, minus 20 capability. Remember, our product is minus 20. Pfizer is minus 70, which is a bigger issue. Yeah. There's actually quite a number of products at minus 20. It's a regular fridge like the one you have in your house. For ice cream, it just has to be validated, of course. But people can store for months the product at minus 20, let's say in big cities or in big warehouse centers. Then they can send it in a regular fridge for 30 days. And so in a lot of low-income countries, it's not an issue. Even I talked to the UNICEF team as we work with them, is there are people doing the last mile, even in coolers and so on, like they do for other vaccines, that are stored, in fridge temperature. And so with minus 20, which is where we picked it, it is not an issue.
So going back to the boost strategy, you probably saw that's quite interesting. So in Europe, they have the problem that a lot of people got the first shot with AstraZeneca or J&J. Well, J&J is single shot, but with AstraZeneca. And now people have been very reluctant to actually get more shots of either one, by the way. So there is that emerging guidelines, I think in France and a couple of other countries, considering heterologous prime boost strategy, where people who have got an AZ potentially would get a booster shot with an mRNA vaccine, right? And so what do you think of that, right? I mean, I know there's a lot of studies being done. I actually haven't seen like recent data, but I'm sure you're all over it. So what do you think of that approach? I mean, presumably it's favorable for you and Pfizer in Europe, but I'm just curious of how you guys look at that internally.
Yeah, so indeed, we were made aware by several governments in Europe where basically they were confronted with an issue, which is people didn't want to come back with a boost of an adenovirus vaccine. And so they're like, what do we do? And so I spoke to a couple of those scientific committees and said, look, we're going to propose to people to get an mRNA boost. Of course, they knew there was no clinical data. And so that's why I'm not recommending it, because we have no clinical data. What we're doing right now for the boost is we're trying a mix of all the vaccines that have been authorized. So as we're preparing the boost, of course, the data we shared last week was boosting people who got the Moderna vaccine last year to simulate, natural situation 6 months or 9 months out. But we are working with the NIH to boost people with our booster strategy that got the Pfizer vaccine first or the J&J vaccine here. We're working in the UK because there's a lot of AZ vaccine for people who got initially the 2 series of AZ vaccine to come with a Moderna variant boost later. And all that data will be available in the summer.
Okay. And you saw that Oxford study where I think it's actually a sizable study, about 830 people where people who got AstraZeneca and then got AstraZeneca, and then a cohort got Pfizer, and a cohort got AZ Pfizer, and they actually saw there was a very material increase of side effects, the usual side effect incidence on the second shot for people who got the adeno mRNA boost on the obviously heterologous prime boost. So I don't know what that means. They didn't give the antibody titers in the study, not at this stage, but does that mean that might correlates with efficacy, or obviously we need to wait for the data, but I thought it was interesting.
Yeah, it's not too surprising because of immunology. I mean, if you think about it, if you give an AZ, ANZ, or an AZ, mRNA, whichever it is, Pfizer or Moderna after, if you look at the data, there's a bit more side effect with the mRNA vaccines, both of them, compared to the adenos. And that's not surprising if you think about the level of antibody titers that you see. And I think that's what we're seeing when you do an AZ first and then an mRNA behind it. Is the second dose is a bit hotter. Immunologists tell you it's a good sign. And when you look at the antibody titer, they are in the sky. And so I think that just there's not, I don't think there's much more than that.
So now talking a little bit about the forward epidemiology, right? And the likely natural history of the pandemic, which obviously will be endemic, hopefully within a few months. How do you see the landscape evolve? So I understand the booster strategy, you guys will be ready and monitoring the variant. But take us beyond 2023. Like, how do you see the epidemiology evolve, over the next, let's say, 3 to 5 years?
Yeah, I think it's going to be similar to what we see with other coronaviruses that most people are not aware of. Like there's one called OC43. It's responsible for around 10% of hospitalization in this country every year. It's a super old coronavirus. It is now believed to be what was called before the Russian flu in the 1890s. And it just come back every couple of years because it has been observed that people that get naturally infected get a 1 to 3 year natural immunity through natural infection. It depends on the age, comorbidity factors, BMI, and things like that, as you would expect. But it just keeps coming back in the community because the natural immunity waned again after 1 to 3 years. Sorry. And so we think it's going to be the same thing with SARS-CoV-2. We're going to live with it. It's going to be around us. What we plan to do about it is to develop a single annual boost, combining a high efficacy flu shot and the variant specific of that year, COVID into the one shot, you go to your CVS or your GP or at your company, wherever you get your flu shot regularly, and you'll get your one shot and you'll have a nice fall and winter. We believe we can really increase the efficacy of the flu. As you know, the flu shot in the good years, they're in the 60%. In the bad year, they're 25, 30%. We believe we could get an annual flu shot in the 90% plus efficacy with mRNA. We're going to be in the clinic very soon, and I'm talking weeks, not months or quarters. And because flu has a provable endpoint, which is HA antibody titer, you can go very, very quickly to authorization. There was not such an endpoint with SARS-CoV-2 because it was a new virus. And so I could see a world in literally a couple of years, potentially as early as 23, where you're going to be to get a flu shot and a COVID shot, both specific to the strain of a moment in your continent in September, and then you're going to have a nice winter.
What is going to be the regulatory path for the booster for you guys and the other mRNA folks?
So FDA has been very clear since the emergence of the South Africa booster. They said studies in hundreds of people, not tens of thousands, and neutralizing antibody, not efficacy. And their thesis basically is to say now there's a lot of data showing that neutralizing antibody is a correlate for efficacy. There's a lot of work being done where people have pulled data from all the different studies because we have a lot of vaccines now. And you can see a very nice correlation where you see the mRNA vaccines at the top, and then you go down, including Chinese, live attenuated vaccine in the 50% efficacy. And so FDA is saying, if you show me the same level of antibody that you show me in your efficacy study, then we'll give you authorization.
So continuing on that page, how do you think about the morbidity risk in the unvaccinated population as some of the variant emerges, particularly in countries where the baseline endemic level might be higher than it is in, let's say, in the US, right? So do we have data already? Can we predict what the morbidity challenge might be for the emerging variant?
We're starting to get some ideas. And I say starting because the data is not that clear, JF, because of which countries have done the work. I would say two countries I will point you to. One is Brazil and the other one is India. It is believed by some antibody work done last year and in the fall that a lot of cities in Brazil got very high natural infection last year for the first wave, with most people asymptomatic. And those people got sick with P1 this winter.
Yeah.
And that's where you see the two factors are at play. I think one is waning immunity with time. Time is not your friend. The farther away you are from either the natural infection or vaccination, your neutralizing antibodies are going one way is down. That's the one problem. The second problem is a variant, which is how far away is a variant from what you saw the last time, whether through infection or vaccination. And this is why I really believe that people should be boosted with variant-specific booster. And we've been saying that for quite a long time. I want my own booster with a South Africa variant because it will protect me because of E40K mutation against P1 and against 351. We believe through preclinical data, which translate through everything we have seen in our 10 vaccine in the clinic in the last 5 years, from preclinical to clinical, also against the Indian variant. We will keep adding more variant as we need as time goes by and as they emerge. I don't think we are done. What I'm personally very worried about is the next 6 months, not in the north. Yeah. We're going to have a quasi-normal summer in the US for sure. It's going to take a few more months in Europe, but in the US, we're going to have a very nice summer. What's going to happen in the south in the next 6 months is going to be pretty traumatic. You see it in Latin America. We don't talk about it a lot in the news, but you see it in Latin America all over the continent right now. There might already be several new mutants in India that nobody is aware of. Because when you have 400,000 people reported infected every day, and you have a 20% infection rate, you have most probably a million or more people that are infected every day. So the chance for virus to evolve is just tremendous. And we always forget that in those countries in the south, you have a very high percentage of people that are immunocompromised with HIV. I was not surprised, given I've done infectious disease for 25 years, to see that P1 and 351 happen in 2 countries with very high rate of HIV. And so I worry deeply about mutants happening and emerging in the next few months, but especially, mutants of mutants. I worried about the mutant of P1 or a mutant of 351, because those are going to come back in the north. Look already in the UK in the last few days. There are some parts of the UK where you see a lot of 617.2, the most, infectious strain of Indian mutant. And the UK is even thinking, oh shit, how it's going to look like? Because, some people got vaccinated back in December and January.
Yeah.
If you think about it, those people are going to have by August, September, 9-10 months of immunity waning. And those are your people at the highest risk, your elderly healthcare worker. And we see cases in India that have been documented, and I've heard through several friends, doctors, people, medical workers that were vaccinated with an adenovirus vaccine back in January, February, that are getting sick with that variant 6 months after.
So for the time being, essentially, you make a pretty compelling case that people will have to be vaccinated at least every year until we get a better understanding of the evolution of variants in the different continent and the different subgroup of population.
Exactly, which is also why, JF, we decided with the board to invest to get 3 billion doses next year, because there is no scenario where we get this virus under control this year at the planet level. But I really believe between 3 billion of us, 3 to 4 billion of Pfizer, their last public number, you could boost the entire planet next year with mRNA vaccine.
Yeah.
Either people who got naturally infected or people who get a vaccination of AZ or J&J or CanSino or pick your vaccine with a highly efficacious, highly specific variant and booster. And what this will do is to slow down the rate of mutation and variant appearing. So I think we really need to nail this virus next year. I think we have all the PCs and all the technology to do it. And then I think, to your point, the pace of mutation will slow down tremendously, will be much more similar than the flu, even maybe less mutation. Some scientists believe that SARS-CoV-2 will evolve slower than flu once in its stable phase, but we're not yet at the stable phase of a virus at the scale of a planet.
So before we I have some question on the screen here, but before I get to that, let's talk about the strategy. Actually, I'd like you to kind of to do something unnatural, to put yourself in the seat of a big pharma executive. Although eventually maybe you'll be in a big pharma seat at Moderna anyway. But what is the vaccine strategy going forward? If you already have an existing infrastructure and very significant material capital investment in, let's say, recombinant or adeno, where do you go from here? Is there some part of the market where you still continue to go recombinant?
So again, that's a decision for those companies to make. I believe, and I think the world, or many people in the world believe now, that mRNA is going to be a big disruption to a vaccine business. And I think in a few ways. One is innovative vaccine where there is nothing on the market. I'm very excited about our CMV, cytomegalovirus vaccine. It's going to get very, very soon into its phase 3. The phase 1 and phase 2 are beautiful. It's a very complex herpes family virus. For 20 years, it has been the number one priority of the National Academy of Science to get a vaccine against CMV. It's the number one cause of birth defect in this country, 1 in 200 child. And plus you have transplant and so on and so forth. It's a very complex virus. We have in our vaccine 6 mRNA molecules, Jeff, coding for 6 different antigens. And the regulators let us go from the beginning, from phase 1, with 6 mRNA molecules in each vial. We have an analytical strategy for it, and we're flying to phase 3. So that's one thing, very complex viruses that have never been addressed. So we're going to grow the pie of the market tremendously. 2 is combination. As I described with our flu, COVID booster, we're also going to add RSV. Some people are going to try to do an RSV single shot. We're going to add RSV to our respiratory panel. We want to have 1 shot you get a year, and you don't get, for most people, hospitalized that winter for respiratory infection. And so I think we're going to disrupt the flu business, and we're going to also create new products like this. And then we want to also go back to existing vaccines. There are still a lot of vaccines on the market today that are okay, not great. So what if you could go and through combinations, through getting higher efficacy, through doing the right biology, go after those vaccines. Another one we're working on is EBV, Epstein-Barr virus. That's the virus driving mononucleosis. And a lot of children lose a lot of precious time in high school, in college because of mono. But another piece that is not as well known, if you look at epidemiology, you have 90% of people that have MS are EBV positive and have had mononucleosis. So we need to run a big study. It's going to cost us $1 billion, and we're going to fund that study, working with governments that have the data to collect the data and so on. But we believe that physics is serious because the numbers are done at the epidemiology level. They are like big country level wide studies. We're going to go like, Merck did with HPV. We're going to go and prove, because that's our scientific physics, that if you vaccinate people against EBV, you're going to drastically reduce the rate of MS in the country. And we're going to go after all of those. I mean, our appetite to invest in vaccinology is almost with no end right now.
Yeah. So that covers actually, I got a few questions on Moderna. So that covers the vaccine part. I mean, obviously, you're developing a massive portfolio here. So not surprisingly, we got quite a few questions on your IO portfolio, including one from our good friend Christian Rommel here. Hi, Christian. Tell us about the IO portfolio and what you're seeing from a data standpoint in terms of your cancer vaccine strategy.
Sure. So on the therapeutic side, and thank you for taking me out of vaccine a little bit, because I spend my days talking about vaccines. On the therapeutic side, we are exploring with mRNA in the clinic a few things. We're exploring, IO with 5 different candidates in the clinic right now. Our personalized cancer vaccine, we can talk back about it again in a sec, our KRAS vaccine, and then 3 intratumoral products, including one partnered with AZ. Sorry. And the idea is not to replace PD-1 or PD-L1. Our idea is, can you combine in an orthogonal way novel biology to an approved checkpoint to improve the response rate of patients? We have no interest in doing another PD-1 antibody. There are way too many of those. And so we're in the clinic. We start to have interesting data. It's still early days. And in oncology, you want to be very, very careful. But we start to have interesting data in our IL-2 program with AZ at the low dose as a monotherapy. We've seen patients that were refractory to Durva, the PD-L1 of AZ, that are showing response to the mRNA IL-2 product. The goal is to, of course, keep pushing the dose up. The first dose we started with was super, super low because the AZ team, as they should be, were very worried about IL-2 because, as we all know, IL-2 is a pretty potent molecule. And so we started piano to make sure to not hurt anybody. And it was a surprise. Nobody expected already some early sign of response at the lowest dose of a study. And as monotherapy, our goal is not to use it as monotherapy. From all our scientific work, we believe it's going to be improving the response rate of checkpoint mono by adding mRNA IL-12 at the same time. We have a couple more, including a cocktail of 3 agents that we're testing in the clinic as well with checkpoints. And then in cardiology, we have 2 programs in cardio that we're very excited about. I think in the last 10 years, there's not been a lot of innovation in cardio. One is our VEGF program that is partnered with AZ, that is in phase 2, where we inject in people's heart within 48 hours after an infarct, mRNA coding for VEGF. It's a one-time intervention. It's 10 to 15 injection at the site of the infarct. It takes around 10 minutes to do the procedure. We have shown in pig models, which, as we know, is pretty predictive to human, an improvement of ejection fraction 6 months after the intervention in pigs. The phase 1 study was very interesting because not only AZ showed the molecule was very, very safe, but it also showed that they had an increase in blood flow at the site of injection, which was the heart, the arm of people in that study. And there was a control on each subject. So it's quite interesting data. The phase 2 is ongoing. I will not be surprised if we have data this year. Then we have an autoimmune portfolio where we're able to deliver mRNA inside immune cells. And we can, with microRNA coding, really dictate, in B cell, T cell, or other lineage of the immune cells. We have a super cool program, which is a PD-L1 protein, not a PD-L1 antibody, As I say, we don't need more of these. But the PD-L1 protein, which is designed as a transmembrane protein, and does an end check with the PD-1 on the T cell. The data looks really interesting. This is something you could not do with a recombinant because it's a membrane-bound PD-1 protein that we make. So we, again, have to see the clinical data. And the last one is rare genetic disease. As we all know, rare genetic disease is, of course, a low biology hurdle to go, give instruction into the cell of a kid to make the protein that they're missing from the DNA of their parents. We're in a clinic with rare genetic disease in the liver. So we're doing basically the opposite of what John and the team is doing at Alnylam. We want to make the protein not turn down an mRNA. And we're making very good progress with Vertex in delivering mRNA into the lung. Through the data I've seen in higher species, in non-human primates, I would not bet against that program. It's starting to look very interesting. So we're going to try it first with CF, where the biology risk is low. If we can show we can, improve disease into those children that don't respond to a small molecule, then we're going to do many more disease into the lung because we think we're getting very close to cracking how to get mRNA into the lung safely to humans.
Okay. Well, Stéphane, we're running out of time. So thanks again for coming today. Really appreciate it. I hope that everybody did. And, keep up the good work. We all need it. Thanks again.
Thanks for being back, Jeff.
viralnews.me